my channel. In this video, I'm going to talk about the concept properties of a genetic code. As you all know, DNA consisting of genetic information which is essential for the synthesis of protein. And these genetic information from the DNA will be first transferred to mRNA, and this mRNA will direct the synthesis of protein. But the question is, how does that simple mRNA consisting of sequence of nucleotide will direct the synthesis of protein? From several experiments, it is concluded that this simple mRNA consisting of genetic information in the form of a genetic code. So, genetic code is a sequence of nucleotide on the mRNA molecule which consisting of genetic information essential for the synthesis of protein. Now, let me discuss about the properties of this genetic code. First one is genetic code is a triplet code. Triplet means 3. So 3 successive nucleotides on the mRNA group together to form a one codon, and that codon codes for a particular amino acid. So each codon codes for a particular amino acid. 4 different types of nucleotide in different combination can form 64 codon, and these 64 codon will codes for 20 amino acid. So each codon consisting of 3 successive nucleotides on the mRNA molecule. Second one is the code is non-overlapping. Non-overlapping means a triplet codon or the sequence of nucleotide on the mRNA is read in a particular order, and each nucleotides can be a part of only one codon. For example, the third nucleotide, that is C, is a part of the first codon, that is GAC, and this C cannot combine with U and A to form another codon, or it cannot overlap. So each nucleotides on the mRNA can be a part of only one codon. Next is the code is comma-less, means there is no punctuation or the comma between the codon, or nucleotide sequence on the mRNA is read in a continuous manner without comma or any punctuation. Next is the code has a polarity, means the nucleotide sequence on the mRNA molecule is read in a particular direction. It always read from 5' to 3' direction, so it has 5' to 3' polarity. Reading the sequence from reverse direction, that is from 3' to 5' direction, may produce different set of protein. Next one, codons and anticodons. Codons are present on the mRNA, anticodons are present on the tRNA. During translation or during protein synthesis, the codons which is present on the mRNA will pair with the complementary anticodon present on the tRNA. For example, as you can see in this diagram, the mRNA has a codon AUG. It pairs with the anticodon UAC which is present on the tRNA. So A pairs with U pairs with A, G pairs with C. Next one, initiation codon. Out of 64 codon, AUG acts as an initiation codon. EUG performs dual function. It codes for methionine and it also acts as an initiation codon. The AUG codon which is present on the mRNA will initiate the translation or protein synthesis, or it is this position where the translation will begin or the protein synthesis will begin. Next is termination codon. Out of 64 codon, 3 codon will not code for any kind of amino acid. That's why they are called as nonsense codon. They are UAG, UGA and UAA. So these 3 codon will not code for any amino acid, and these codon will not have anticodon on the tRNA, because of which the presence of this codon on the mRNA will terminate the translation, or it will stop the translation process. Next one, the code is degenerate. As I already told, out of 64 codon, 61 codon will code for 20 amino acid. From this, it is clear that some of the amino acid is coded by more than one codon. For example, the amino acid leucine is coded by 6 codon like UUA, UUG, CUU, CUC, CUA, CUG. These 6 codon codes for the amino acid leucine. Next one, the code is unambiguous, means one codon will code for one particular amino acid and it always codes for the same amino acid. Next one, the code is universal. As I already told, a codon codes for a particular amino acid and it remains same in almost all organisms and in all places. For example, the codon UUU code for phenylalanine in the case of human being, and even in the case of bacteria, UUU code for phenylalanine. There are some exceptions like mitochondrial codon in some of the protozoa. Next one is Abel hypothesis, which is proposed by Crick during 1965. According to this hypothesis, the first 2 bases of the codon on the mRNA has precise pairing with the anticodons on the tRNA, or it is purely based on the complementarity. Whereas the pairing between the third base of the codon with the anticodon is less specific, or it may not be based on the complementarity. As you can see in this diagram, the codon CUC pairs with the anticodon GAG in the normal pairing, which is purely based on the complementarity. C pairs with G, U pairs with A, C pairs with G. Whereas in the case of Wobble pairing, the codon CUU pairs with the anticodon GAG. The first two nucleotide, that is G pairs with C and A pairs with U. It is purely based on the complementarity. If you observe the third position, G is paired with the U here. Instead of pairing with C, here G is paired with the U. So it is not based on the complementarity, or pairing is less specific at the third position. So that is nothing but the Wobble pairing. This Wobble pairing will permit the tRNA to combine with more than one codon. So that is the benefit of Wobble hypothesis. So that is all about the properties of a genetic code. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.